The extended synthesis is the name for a whole range of new proposals that have been made by leading evolutionary biologists who, on the one hand, recognize that neo-Darwinism is inadequate, that the mutation natural selection mechanism does not generate new form and new biological information. And on the other, biologists who want to hold on to a strictly materialistic or naturalistic approach to solving the problem. So they're proposing other materialistic evolutionary mechanisms to try to supplement the perceived and increasingly agreed limitations or liabilities of the mutation selection mechanism. So the extended synthesis the terminology is based on, it's a variation on the old idea that neo-Darwinism was a synthesis of Mendelian genetics and classical Darwinian theory. And that in the 1930s and 40s was called the new synthesis. So the extended synthesis is new evolutionary mechanisms that are being kind of bolted on to the old neo-Darwinian theory. But the problem we've found with each of these new proposals is that they don't account for the origin of the information necessary to build new forms of life either. And typically they end up either not addressing the problem of biological information, or they simply, or they will end up begging the question. They'll explain the origin of new form by presupposing pre-existing unexplained sources of information. Let me give you an example. There's a brilliant scientist at the University of Chicago named James Shapiro. He gave one of the best talks at the conference in November of 2016 at the Royal Society in London.
So we return to our conclusions. The only constant in life is change at all time scales. To deal with change, cells read their genomes dynamically, and that allows them to adapt to changing conditions both during cell reproduction and multicellular development. And genome reading is a function of prior inscriptions, which, as we have seen, is a function of prior natural genetic engineering processes, which have incorporated coding data, signals for interaction with cell molecules and transcription and replication, the formation of nucleoprotein complexes, and chromatin formatting. Genome inscription is an active physiological process at all biological timescales. It is subject to regulation, to being activated and inactivated, to being targeted within the genome, and we have yet to explore how extensive that regulation is. And finally, we know that in evolutionary history, cells have actively written data and formatting signals into their genomes by the processes of symbiogenesis, horizontal transfer, and natural genetic engineering. Thank you.
He has a new model he calls natural genetic engineering. And what he points out is that the mutations that we see taking place in nature very often are not random at all. They're not random with respect to the survival needs of the organism. Rather, they are in some way produced by guided mechanisms that are reflect a kind of pre-programmed adaptive capacity. So if an organism is under some sort of environmental stress, that triggers a response that either expresses some pre-existing genetic information or ramps up mutational processes in certain designated parts of the genome. And these are not random responses at all. They're under what he calls algorithmic control. So there's information already in the cell that's telling the cell how to respond to these environmental stressors. And what we want to know is, where does that information come from?
Evolutionary biology is a thriving field of science. Ideas are flooding in from other fields such as genomics, epigenetics, developmental biology, and ecology. Innovation and thoughtful reflection is a natural aspect of science, and that's what the extended evolutionary synthesis is all about. We want to help build an integrative evolutionary framework. We believe that researchers in the sciences, social sciences, and humanities can all play a part. The extended evolutionary synthesis is a new way to think about and understand evolution. It extends traditional theory by recognizing a broader set of causes. Knowledge of how organisms develop, grow, and interact with their environment helps scientists to account for both the diversity of life and the processes of adaptation. We're interested in the idea that the way organisms develop shapes the direction and rate of their evolution, and that inheritance occurs not just through the transmission of genes, but through many additional means, including the developmental conditions that parents construct for their offspring. The extended evolutionary synthesis is more than just a different way of thinking about evolution, however. It's an active research program. These topics have become particularly important because they highlight the differences in how researchers understand evolution. They are the disputed territory of contemporary biology, lying on an intellectual fault line that divides evolutionary biologists. The extended evolutionary synthesis seeks to get to the bottom of these differences.